Yeah. Can you just say me if you hear something, if you are online? Someone? Yeah, thank you very much. Here we go. Today, if I remember, we're gonna talk about mitochondria. That's it. So we have three groups, and here again, if I remember, we are going to start by a presentation of the review. Right? That's it. Who is in charge of that? Mine, maybe some people. So we have three papers. One is a review, and the other one are articles.
Yes, yeah. I think it's us.
Hélène, yeah. So if you can just share your presentation, if it's you that is in charge of the presentation of the review, we're gonna start and ask a question about the role and the consideration of mitochondria as maybe potential biomarker of aging in a context of mitochondrial dysfunction, of course. And we can extend the discussion on different things such as oxygen species release, or even other stuff that are really important and crucial for understanding the role of mitochondria. Okay, so let's start by your presentation, and then we're going to discuss about this first presentation, and with the article, we're going to see exactly where we are. Okay, so the floor is yours. You can start whenever you want.
Sorry, yes. So you hear me? So we all know that the mitochondria play a role in ATP production, protein maturation and degradation, as well as in the regulation of apoptosis. So these organelles have their own mitochondrial DNA, which is especially vulnerable to oxidative damage due to its proximity to the respiratory chain, the main source of reactive oxygen species. So with age, mitochondrial function tends to deteriorate, a phenomenon largely attributed to the accumulation of oxidative damage over time, which affects the structure and function of mitochondrial DNA, as well as mitochondrial proteins and lipids. So the accumulation of damage can lead to reduced oxidative capacity, the decrease of ATP production, and also the increase of ROS production. And this creating a vicious cycle of mitochondrial dysfunction. So this review explores the primary functional and structural changes in mitochondria associated with aging, and their impact on cellular function and the importance of mitochondrial maintenance processes to better understand the mechanism underlying aging and identify potential strategies to delay its effects. So the first point is changes in mitochondrial DNA in aging. And so we are going to talk about the mitochondrial theory of aging. So this theory suggests that mitochondrial DNA has a higher mutation rate and processes a less efficient repair machinery. So this accumulates the mutation. And so the accumulation can affect the respiratory chain by dysfunction and by disrupting oxidative phosphorylation. And this can lead to mitochondrial dysfunction and accelerated ROS generation. So experiments have validated the observation of the dysfunctional mitochondrial and by the increase of ROS generation by with these mutated mice with the defective mitochondrial polymerases that accumulate more mutation and show signs of premature aging. However, the data shows that while mitochondrial DNA mutation accumulate linearly over time, there is no significant increase in ROS production or antioxidant enzyme activity, challenging the vicious cycle theory associated with aging. Additionally, the proximity of mitochondrial DNA to ROS production sites in the respiratory chain and its lack of histone make it particularly vulnerable to oxidative damages. In rat hepatocytes, this molecule, which is a marker of DNA oxidative damage, is 16 times higher in the mitochondrial DNA than in nuclear DNA. And also in the skeletal muscles and liver of rats, we can see a decrease of mitochondrial DNA copy number. And that suggests that aging affects mitochondrial DNA quantity and integrity. Then we're going to talk about ROS prediction. And so we know that ROS are toxic, but it also can play a role in signaling. So, low ROS levels may extend lifespan, as we can observe in C. elegans, where a slight ROS increase activates the HIF1 transcription factor that is linked to longevity. And also moderate inhibition of mitochondrial respiration has been shown to increase lifespan across multiple species, suggesting an evolutionary conserved effect. Antioxidant enzymes such as superoxide dismutase and catalase protect against oxidative stress, and genetically modified mice overexpressing these enzymes demonstrate increased longevity, whereas the absence of superoxide dismutase leads to premature aging and mitochondrial dysfunction. Likewise, mice deficient in this protein that generates ROS independently of respiratory chain show an increased resistance to oxidative stress and 30% lifespan extension. So with age, ATP production declines with an 8% decrease per decade in human. Elderly individuals also display reduced oxidative capacity per mitochondrial and muscle volume. However, research suggests that mitochondrial function decline may be influenced by reduced physical and activity rather than aging alone, as no significant correlation between age, mitochondrial respiration, and ATP flux was observed after adjusting for physical activity levels. Then we're going to talk about the point of age-dependent changes in mitochondrial dynamics. So the mitochondrial dynamics, which encompass movements along the skeleton, regulation of architecture, and fission-fusion events, are crucial for maintaining mitochondrial functions and play a role in aging. As individuals age, there is a progressive reduction with mitochondrial density observed in skeletal muscles. suggesting a decline in mitochondrial biogenesis, potentially linked to decreased levels of PGC1 alpha, which is a key regulator of biogenesis. And studies in mice have shown that overexpression of PGC1 alpha can reduce sarcopenia and enhance mitochondrial function, indicating its protective role. Aging also leads to an imbalance between mitochondrial fission and fusion. Fission is essential for eliminating dysfunctional mitochondria through mitophagy, helping to prevent accumulation of damaged mitochondria. However, with age, there is a decline in mitophagy, contributing to the buildup and impair mitochondria, increasing oxidative stress and being associated with higher rates of apoptosis.
Now we are going to talk about another mitochondrial function that have a role in aging. It is true that aging leads to increased apoptosis, and in particular with mitochondrial way. In fact, this apoptosis is induced in response to pro-apoptotic stimuli or in the case of severe failure in oxidative phosphorylation. As you just saw before, if the mitochondrial dynamics is dysfunctional, you can have severe failure in oxidative phosphorylation. But we are the paper are interested about pro-apoptotic stimuli. And the apoptosis, there are two pathways about this apoptosis. So you have the caspase-dependent apoptosis or the caspase-independent apoptosis. As you can understand with the name, the caspase-dependent apoptosis is the induce of apoptosis thanks to caspase. Rather, the caspase-independent apoptosis is the induce of apoptose with a pro-apoptotic stimuli such as endonuclease G and apoptosis-induced factor. So the question that we that the paper are interested is which apoptotic pathway is implicated in age. So it is sure that apoptosis increase significantly with age, and the result that they observe in other papers is that there is only a significant upregulation of caspase-independent pro-apoptotic pathway in age rats and early people, and no significant changes in the expression of caspase. So we can say that the apoptosis increase but tends to take the caspase-independent pathway, and that suggests that there is a different aspect of apoptosis regulation in age individuals. Another mitochondrial function is highlight in aging, indeed, the mitochondrial quality control. In fact, mitochondria have a number of specific enzymes such as protease to refold and eliminate misfold proteins. In fact, mitochondria contribute to the cellular system of the protein quality control associated with ubiquitination and protease-dependent degradation of unfollowed protein. To illustrate that, they in other paper, there is an experience with the depletion or the downregulation of mitochondrial protease such as LON and PALON1 that have a role in quality control or in degradation of oxidized protein. And when they deplete or downregulate this protease, we can see dysregulation in quality control and degradation that contributes to aging and age-related disease. However, we know that with aging, ubiquitin-specific protease and certain proteasome subunits are upregulated, while other components are unchanged or decreased. For example, in muscles of age rats, the expression of proteasome-associated protein was increased, and level of some protease are higher in age rats compared to adult animals. So they can conclude that age-dependent activation of the ubiquitin protease system may contribute to enhanced degradation of myofibrillar protein and age-related muscle atrophy. Now we understand a lot of mitochondrial function in aging. So in the paper, they highlight the role of dysfunctional mitochondria in aging disease, and in particular in vascular aging and in the development of atherosclerosis. In fact, the probability to develop this disease increase with age, and that's why it can be considered as a disease of aging. And in the paper, they highlight the potential link between mitochondrial health and vascular integrity. So what we know? We know that there is a number of diseases that are associated with somatic mutation in the mitochondrial genome. That leads to develop, to increase, the oxidative stress, and also that can lead cardiovascular disease. We know also that there is an association of the mitochondrial DNA mutation and the severity of atherosclerosis. In other papers, they show that some mitochondrial DNA mutation are associated with this disease. And also, we know that we have structural changes in mitochondria in this disease. So to understand more, you have here electron microscopy images of ultrastructural appearance of mitochondria in human aortic. You have in A and B the health mitochondria, and in the C, D, E, and F, you have the mitochondria in atherosclerosis lesion. As you can observe, we have an alteration such as a deformation of the cristae. So it is the fold created by the inner membrane of the mitochondria. And also, we can see with the arrow the appearance of vascular structures. So the hypothesis of the paper, thanks to these 3 points, is that maybe a link between the structural variation in the appearance of mitochondria and the somatic mutation in the human mitochondrial genome, and that it can be determinant to understand more the pathology the pathology arteriosclerosis. So to respond to this hypothesis, they measure the levels of mitochondrial DNA of mutation in different types of this disease. And they saw that we have an homogeneity of an affect. No, they saw they compare homogeneity of unaffected aortic intima and lipofibrous plaques of human aortas. And in this and they see they saw that there is an average level of heteroplasmy for some mutation of the mitochondrial DNA in this pathology. And they also saw that there is a significantly higher prevalence and mean value in fibrous plaques as compared to non-xerotic enzyma. So that can be a mark that thanks. Suggesting that there is a more marker in disease tissue, which may indicate a causal role in the development of arteriosclerosis lesion. So the perspective for this correlation between somatic mutation and the structural aspect of dysfunctional mitochondria can be more learned, but we know that somatic mutation may play a role in the development of this pathology, and they also suggest that heteroplasmy could potentially serve as a biomarker to assess the risk of developing arteriosclerosis. To conclude, we can say that the role of mitochondria in aging is very complex because with the multiple changes in mitochondrial function, structure, distribution, dynamics, we can obtain aging or age-related futures. Also, the studies in model organisms have shown that both suppression and stimulation of mitochondrial function can extend lifespan. For example, downregulation of M motor signaling associated with enhanced protein synthesis and cell growth was shown to increase longevity in model organisms through improved mitochondrial efficiency and better energy consumption. Also, control oxidative stress such that induced by a slight increase in ROS could paradoxically promote longevity. This suggests that optimal redox balance rather than absolute elimination of ROS could be beneficial for mitochondrial health and longevity. Moreover, caloric restriction is emerging as a powerful intervention to improve mitochondrial biogenesis and function. In particular, it activates molecular pathways such as SIRT1 and PGC1 alpha, which stimulate the production of new mitochondria and optimize cellular respiration. So it can be, but the mitochondria, the function of mitochondria in aging still not very understood. So we need to do other experiments and maybe to refer information from more paper that are more young. Thanks.
We don't know if you have the next slide or not. So, okay, thank you very much, Hélène. Thank you very much, Sophie. So we started by the review. It's maybe more difficult to ask a question, but it's also very interesting for you to try to maybe discuss around what has been evoked here. So first of all, I have a question, not only for you, Sophie and Hélène, but also for the audience. Mariam, we are going to ask you a question. Don't worry. The first one is, they propose, I think that it was Sophie proposed, to consider DNA mutation and particularly mitochondrial DNA mutation as a kind of biomarker of aging. What is your opinion? What do you think about that? And regarding the presentation you just made, what can be proposed in the future as a kind of biomarker regarding mitochondrial function, mutation, etc.? What is for you, if you have $1 million to put on this kind of study, what is for you the focus you should have or you would have? You can also answer, of course, Hélène and Sophie, but I would like to all the platform. But it's maybe more interesting, more easy to do. There is no answer, particular answer. I just want to have you to get your point of view. What do you put your interest? No interest in mitochondria? What is your opinion? They talk about different things. First of all, they talk about genetic. We're going to come back on that. Then they talk about structure, and finally, also they talk about function. What do you think it should be more appropriate to create or to consider as a biomarker? How do you plan to do that? What kind of tissue collection or fluid collection do you want to do? Because you know what is mitochondria. In your university courses, I think that you talk about this organelle very often. What is your point of view regarding aging? Sophie, Hélène, do you have an idea? Now you present the overview. What do you think about that? What do you think about considering mitochondria as a biomarker? What is the mainstream that we can favorize to develop some studies? You still with us?
Yes,
Hélène?
yes.
Yeah, so
maybe we can, as with the atherosclerosis disease, we can mesurate dysfunctional mitochondria we can understand more about mitochondria that are dysfunctional in disease to maybe find therapies or to
yeah, but as I understand the idea to consider mitochondria as target of biotherapies, maybe in the future we're going to also discuss about that at the end of today. But my question was regarding the biomarker of aging. you present different part of mitochondrial role or mitochondrial feature that can be attributed to aging processes. According to you, and here again, according to the audience, what should be the more interesting thing, the most interesting thing to favorize or to study in order to get an idea about the relationship between mitochondria and aging process? Do you focus on the DNA? Do you focus on function, etc., etc.? What do you? It's just a discussion. Yeah.
Production of ROS.
Yeah, ROS production. How do you measure that? How can we measure? Do we know for the ROS production? You know how we can measure the ROS production? Imagine that you have the possibility to collect some skin biopsy for the patient, from the patient. And Mariam, you can open your microphone and maybe participate. It's better for me than writing something. If you have to interrupt us, don't worry. But how do you imagine that in the future? You want to manage the capacity of mitochondria to produce ROS in your patient. What is your process? Yeah.
Maybe we can measure the enzyme activity, like the
Yeah, like they propose. So the antioxidative enzyme, such as sodium oxidase synthase or maybe catalase or some stuff like that. This is something that is already a little bit done, as you probably know. When we can make some analysis, this is something that we are measuring. It is for you, according to you, is it? Sorry, Mariam. Is it something that is directly linked to the capacity of mitochondria to produce oxygen spaces, or it is an indirect measure? My question, yeah?
Indirectly.
Yeah, so that means that you are trying to measure a kind of the capacity of adaptation of this enzyme to adapt, of course, to the amount of ROS. But this is not a direct measure. Do we know how to directly measure the ROS production? Did you already do that, or maybe did you already see that in the publication? Of course, yes. But how can we do? There are some dyes, you should know that, no? There are some dyes that are able to label, you know, the ROS production. Of course, you need histology slice. So at the end, that means that maybe you need to also to make some tissue collection. If you have some biopsy from, for instance, from skin, you will be able to analyze the ROS production. So this is here a direct methodology. You don't need to have fresh tissue. That's very surprising, but you don't need, because you are finally, by this kind of dye, you are directly measuring the enzymes that are producing the ROS and not the enzymes that are trying to, of course, to regulate the ROS. It's interesting, but it's not something that is easy to do in clinics. So what are your other propositions? I guess that I have another question or another suggestion, maybe. Okay, that's also another point about the structure. We're going to open the discussion about the structure. I'm going to come back on that. That's a very good point. Well, I have tons of questions, but I would like maybe first to let the audience speaking. Do you have any question? Do you have any commentary? Yes.
Can we measure the ROS production by flow cytometry?
Yeah, we can. This is a question for me or for Hélène and Sophie, I guess. For me, yeah. Can we measure the ROS production by flow cytometry? I don't know. Is it a question or this is something that you maybe already know?
I saw it in course.
And which kind of because you know flow cytometry is based on antigen and antibody relationship.
With the fluorescent dye.
Yeah.
It's called ECFH.
Yeah.
And it can measure the level of ROS in the population.
Okay. So this is something that could be done maybe also on blood cells. So that could be very informative. Remember that what we discussed on last Monday, to find some biomarkers that are easy to manage. So that means that you should find some biomarker of aging in the future that are maybe retrieved in blood cells, in blood, in fluids, whatever, in saliva, urine, so whatever. I mean, that the best should be something like that. Of course, if you must do a kind of biopsy or something like that, this is more difficult. Just to say that your hygiene process is ongoing, okay, to a patient. The patient accepts the biopsy when the risk is high. If you just to measure a very slow and progressive process and say, okay, you are aging very fast, I'm not sure they accept. Of course, they can accept in a research context, but when you are at the hospital, they don't accept to make some biopsy just for that. So that means that you need to make or to envision your biomarker on different tissue collections, such as maybe blood cell, or maybe swab, as I told you last Monday. Another remark, another question regarding this presentation. So it was a review about mitochondria. Of course, we start by the different pillar. What do you think about that? Do you think that mitochondria should be something that we need to manage in the future as a marker of aging regarding the presentation they made? No? What is your opinion? I'm going to just to read the question raised by Mariam, but you should have an opinion. If not, okay, Mariam, so I'm going to ask you a question to Hélène and Sophie. If lack of ROS is beneficial, so it was one of my questions. So how does it theory account for contradictory findings, such as lack of lifespan extension in certain antioxidant study? The ROS, it has been also said, which can fetch sample from different age group. You can fetch sample and then see whether some specific mutation associated. Okay, and the lab. What do you think about that, Sophie and Hélène? The discussion around the ROS production. So you mentioned during your presentation that ROS are necessary, and finally you said that ROS are detrimental for cells, etc. What is your opinion after reading the review, and maybe after thinking about that to make the presentation? What would you say about generation of ROS?
Well, the first like the theory, that was the theory that said that ROS is responsible of damages and dysfunctional mitochondria. And I think it's the, I don't know, I think that well, we don't have like exactly results, but the results shows that it's not the only. I think it's, well, I don't know. I think we need like some more experience on mitochondria and ROS, because it's shown that it maybe it's like a misunderstanding of results, and maybe mitochondria don't like ROS don't really play a role on the function of mitochondria.
Okay. So the other, do you have an idea about that, an opinion? Yeah.
Maybe the problem is not the production of ROS who are essential, but the destruction after the use of ROS.
Yeah. So did you hear about something called the mitochondrial theory of aging? All of you, or never? You never heard about that? You know, the mitochondrial theory of aging is based on what we are discussing right now. It's based on the fact that mitochondria are generating ROS. This is something that we know. Okay. Right now, in your cells, mitochondria are generating ROS. Okay. Around something like I don't know, I don't have a quantification, but this is something that is really well studied and well known. Okay, this is physiological. Unfortunately, as we just discussed, the imbalance, the balance is lost during aging. Why? Two reasons. First of all, because the mitochondria is producing more ROS, and the second one, on the other hand, as it has just been proposed, because of the antioxidant system are altered. Okay, so taking together these two arms are going to lead to what is the final idea. It's an accumulation of ROS. All is about quantity. Okay, all is about quantity. In physiology, the quantity is quite low and is even necessary for leading some different processes. For instance, last time, I don't know if you remember, but on the whiteboard, I draw an arteria with the endothelial cells and the small muscle cells. The nitric oxide is a rust. Okay, the nitric oxide is a rust, but it's necessary for vasodilatation. It's also necessary, the same molecule is necessary, for instance, for glucose entry into the cells. Okay, so we know that at low quantity. When this quantity is too high, of course, and when ROS are going to accumulate, what is the first target of this ROS that could account for an acceleration of aging process, according to you? Because I don't know if you got it through the presentation. What is the first target within the cell for the ROS that could explain why ROS are strongly involved in aging process, according to you? Come on. What is the first target of the ROS within the cell that could explain how we can link ROS and aging? What is the consequence of accumulation of ROS in the cell? Sorry?
Oxidative stress.
Sorry?
Oxidative stress.
What is it? What is oxidative stress? Or can you explain what is oxidative stress? What we call about stress? Oxidative, we know. You know why we say that it is oxidative stress? Because ROS are coming from the oxidation of the molecule, okay? So like, you know, when you drive a car, of course, you have some gas that are emitted. Here, the same, okay? But the stress, all is in the stress. The consequence of ROS accumulation is, so that the last consequence, but DNA damage.
Activation of pro-inflammatory.
And also pro-inflammatory process. But the first one, the one that we are just discussing right now is what? Is DNA damage. These molecules are very highly reactive with DNA. So what is the first DNA that is going to be affected by this ROS? Mitochondrial DNA. So that's why we know that if there are too much ROS within the mitochondria, and particularly within the cell, even in the cytoplasm, the first consequence is the degradation and alteration of mitochondrial DNA. That's why we are talking about, finally, a kind of intrinsic capacity of ROS to alter the mitochondrial DNA. And the second one is the genomic DNA. Okay, the genomic DNA will be altered by the accumulation of ROS, and as a consequence, you know, and we are going to talk about that, I think, tomorrow or on November the 14th. There will be, of course, the activation of senescent mechanism. Okay, so the aging process is like that. Between these different steps, of course, if the amount of ROS production is quite high or low, there are other parameters that can be affected, such as pro-inflammatory cytokine production, etc. But you have to understand what we are going to talk about. Okay, okay, this is the first step. Now, second part. Well, I have a question before switching to the other part to Hélène and Sophie. Hélène and Sophie, do we know something about? But I just evoked the answer, I guess. I wanted to know if we know something and if they discuss something about the capacity of ROS to target mitochondrial genomic DNA. You know, if we know something about the mechanism that occur for mitochondria and genomic DNA alteration regarding the ROS production. Do we know something or no? Hélène and Sophie.
I think no. No. It's not a very. If you take a look in the paper, in our paper, no. But if you want to very how can I say that? Okay, it is a the finding that we have, it's come from other papers. So if you want to see that, maybe it is on other paper, because it is the conclusion of these papers on our paper. I don't know, it is very clear. So in our paper, no, we don't have the.
Okay, in the review, they never talk about that. They never
Yeah,
about the
exactly. Yeah.
hyperreactivity of such DNA compared to the other one. But as I just explained to you, that's why I said that I told you, I mean, that I evoked the answer before asking the question. The hypothesis is that mitochondria generation of Ross is first affecting mitochondrial DNA because of the location. That's it at the beginning. And then, of course, secondarily, you have also the genomic DNA that it could be affected. But the first one, so the more early, is the mitochondrial DNA that is affected. If you don't find any mutation in mitochondrial DNA, there is no reason to consider that the work production should affect the genomic DNA. Do you think that studying the mitochondrial DNA is something that we are doing in routine in clinics or even in the lab? Why? I say some face saying no, but why do you think it's not something that we are doing? Why we are not doing that?
Because of ethic.
Well, we can measure we can do that sometimes. I never seen opposition from the patient or something like that. This is not due to the ethics stuff. What is the main reason? Do you know who? Yeah.
I think that measuring the mitochondrial DNA is not don't have an impact in the clinics, but in researchers.
Why there is no impact on clinics? Because today we're asking the question if we can consider the feature of mitochondrial DNA mutation as a biomarker of aging. You said that, Farah said just, no, it is not interesting because, but because what?
Because I think that mitochondria as a biomarker of aging, will serve as a preventer more than.
Yeah, it can be also a kind of diagnostic tool. If your DNA is totally mutated, that could account also for maybe explaining some pathologies and some acceleration of aging process. I mean that the answer is not on the fact that this is interesting or not, but this is doable or not. What do you need to make this kind of measure? What do you need?
Like maybe because it's not specific to a certain, like it's specific to an individual. So it's more, if I want to say, specialized.
Can you say it again, because we didn't hear very well. Zaina, I guess. No? Zaina, you are here? There is someone that is called Zaina in the care. No? Okay. Why? The question is why? Oh, sorry. The question is how can we measure that?
By biopsy. No?
Okay. Let's say that we have some cells from a donor, whatever the cell we consider, the blood, the swab, whatever we want. How can you measure that? How can you measure DNA mutation? Come on. What we need to do? Yeah?
By using process of
Yeah.
two different, in this case, mice, for example.
No, we are talking about things. So I think that I give you some cells from whatever, from skin biopsy if you want. And I ask you measure my mitochondrial DNA mutation. What would you do?
Sequencing.
What do you say?
Sequence at the end.
Yeah, so we are going to make some DNA sequencing first. But before, what we need, if we do sequencing, if you do DNA sequencing on the bulk, on the cells, you will have the genomic and the mitochondrial one. It's difficult to decipher. So what we need to do first? No, what we need to do first? The question is how we can get an idea about the DNA mutation of mitochondria. You need first to
Linear DNA.
So isolate the mitochondria, and then you will make the sequencing, and then stop that because it's not enough. You have the sequence of my mitochondrial DNA. you need to find the mutation, so to try to align and to find. So it's a very long-term process, very expensive and very difficult to do. So that's why today, this is not something that is done in clinics and in routine, of course. Maybe in the future, we will find some marker, better indicator, that says that, okay, the DNA is mutated by different dyes. We can say that the DNA, particularly the mitochondria one, is mutated, okay? Some of my colleagues in my institute do that. In a fish model, it's quite difficult because, of course, the sequencing is not so easy, and then we are talking about a small DNA. Okay, so it's not so easy. Okay, what about genetics? What else about genetics? So Hélène and Sophie present you the role of different mutation. We talk about that. How can we explain, or maybe do we have an idea, Sophie and Hélène, about how we can explain that this DNA is not repaired? I mean that in my opinion or in my mind, since I'm a student, they teach me that the DNA should repair. What do we know about reparation process in mitochondrial DNA? Did you see something or no? Hélène and Sophie?
Yeah, yeah, I only see in the paper that the DNA repair in mitochondria is, well, less effective, but I don't really know why.
Well, that what we know, of course, is that in mitochondria, and particularly so in mitochondria of H people, the proteins that are able to repair the DNA are less efficient than, first of all, for the genomic DNA, and then, of course, in a young condition. Okay, so we know that there is an alteration of enzyme or protein like PAR that are able to repair the DNA after mutation. So that's why we can encounter more mutation in the mitochondrial DNA associated with age than to compare that with young people. Okay, so that could be also a kind of not a marker, but a kind of index that shows you that we can measure the capacity of mitochondrial reparation regarding finally the occurrence of mutations. Okay, so you can understand that this is something doable, measuring mitochondrial DNA alteration by measuring different things. The origin of this mutation, we talk about the loss, but also the consequence of this mutation. Okay, this is something that we can do, but nowadays this is not something which is done in routine by different bias, such as we just evoked. Okay, so first of all, we talk about genetic and mitochondria. Secondly, we consider that this genetic alteration may affect the mitochondria itself. Hélène and Sophie present you some feature about this alteration. And we talk about structure. What do we retain about the structural alteration related to aging for mitochondria? What do you want to retain? They present different things. What is the thing that you have in your mind? Yeah?
The formation of the vacuole.
Okay, we have, they about that, or this is something that you already did? They talk about that, maybe I was writing something. Okay, formation of some vacuole. Maybe Sophie and Hélène, can you say it again and explain us what are these vacuole and what is the interpretation of the formation of this vacuole within the mitochondria?
Yes, so the vacuole.
I don't remember where exactly you talk about that. I didn't even...
Hélène, can you go to the... Encore? Merci. Non. Voilà. Okay. So in the CD, in all the four images in down, you can see the appearance of vacuolar structure inside the mitochondria.
Can you, the C and the D, that's it?
C, D, E, F, sorry.
Okay, C, D, E, and F. Okay.
And
this,
also in these images, you have the deformation of the crystal. I don't know. I
yeah,
can.
well, we are going to talk about the criteria after, but first of all, we
Okay.
want just to talk about the apparition of this vacuole. Can you just explain us what is the hypothesis about this vacuole? Why they are here and what is the difference?
It's like a structure in zone of
In?
odematous matrix of mitochondria.
Yeah, okay, but why we talk about vacuole? Do we know about what about the content of this I don't know, vacuole of this new structure or what?
I don't know.
No? You didn't see something about that? What is your opinion, the others? Why we have accumulation of vacuole within this mitochondria when we age? Yeah?
I think it's because of permeability of the mitochondrial membrane.
Well, yes, some people say that maybe, of course, because you can see that the mitochondrial integrity is not something that is really correct right now. What is the other perspective? What is the other proposition? Permeability? And then, what is the role of mitochondria? Don't forget it. What is the main role of mitochondria?
ATP.
Producing energy from what? No, producing energy, from producing ATP from what? Not from oxygen, I mean that's from substrates such as glucose and lipids. So that means that the mitochondria, you should maybe hear about this kind of protein which is called CPT1, carnitine palmitoyltransferase 1. The mitochondria own different proteins here on the outer membranes that are able to make a kind of flip-flop between the outside and inside the mitochondria. So promoting what? Promoting the entry, of course, of lipids. But if these lipids are not used, they are going to be a kind of store. This is not exactly storing, but this is an uncontrolled storing to this kind of vacuole. Okay, so some explanation may reside in the fact that a poor use of these lipids by this outer mitochondria, the permeability which is also altered. Okay, what else in terms of structure? We talk about vacuole. What did you notice that maybe is really important if you have to consider the mitochondria of an age people? What would you maybe focus on?
Fusion and fission.
Okay, we talk about fusion and fission because Helen and Sophie present us a very interesting scheme showing what is exactly fusion and fission. Did you read about that before? Or we need to come back? Okay, so everybody knows what is fusion and fission. First of all, before discussing about that, fusion and fission, you are used to consider mitochondria as maybe very simple and individual organelle. This is not the case at all. Not at all, okay? Mitochondria are not isolated like that, okay? We think that is the case because when we make a kind of slice, of course, we have the feeling that mitochondria is isolated, but this is not the case. Mitochondria are organized in network. And this is very impressive to see that in 3Ds, of course. But what we know is that this networking is something that is mandatory and essential for the mitochondrial functioning. Okay? Why I'm talking about that? This is very recent result, but also very interesting because some people are showing, it is not published yet, but some people are showing that with age, the networking is totally lost. Mitochondria are still there, but the networking is lost. I don't have the opportunity to show you because these are confidential results and I learned from a congress, but I can maybe draw you some stuff that I have difficulty because with this. We have a kind of piece of tissues. It was muscle, skeletal muscle. Sorry for those that are online. I'm going to show you after a kind of picture of my draw. And this is a 3D reconstruction. And how can I show you that? You have the different, this is muscle, so you have the different fibers here. Yeah, this is of course badly drawn, but it's not the most important. Okay. you have the fibers that could be something like that, okay, and like that. So this is a very small tube, a very small biopsy, something like 1 millimeter by 1 millimeter, and you can see the different fibers that are like that. And the skeletal muscle should be something like, okay, like that. You understand what I mean? Okay, a very small fibers. When you are managing the mitochondria, you don't have stuff like that. This is not the case. This is not isolated. What we saw is some stuff with a networking between the mitochondria, which is essential, but clearly essential. And you can see this kind of thing. And very interestingly, if you are measuring, if you are doing the same experiment and the same 3D measure in, and here, of course, you can see that here also. Okay. If you do that in H people, you will find that the networking is lost. We don't know exactly why. We can identify the different mitochondria, but the networking is lost. So the most important thing, not the most important thing, but one of the important things before starting and before discussing about mitochondrial structure is the networking first. And why we are not aware about that, why we never discuss about that, because of technology. Now the technology are able to show us that. Okay. What about structure? What about size and shape? If I remember, Sophie, you tell us that during aging we can see less mitochondria, and you didn't talk about the shape. Can you say a word about the shape? What do we know about mitochondrial shape? You know what is shape?
During dynamics or?
No, during aging. Because if I remember, I know that you mentioned that during aging there is a decrease of mitochondrial number. That's it. So this is something that we know. Decrease of mitochondrial number. What can you say about shape? Did you see something about shape of mitochondria?
No, it's more focus on the dysfunctional mitochondria shape with aging, or just the shape.
Okay. The others, what do we know? What do you know in the audience about shape of mitochondria? What is your feelings? When we age, we have less mitochondria, and they are what? Yeah, this is a function. This is a third point, and they are what? Bigger, smaller? Who is for bigger? Okay. Who is for smaller? Okay, so we have 90% for smaller. Actually, this is something I was thinking about also in something like maybe 5 years ago. When you read the literature about mitochondria and about aging, you will find a lot of evidence in showing that mitochondria are smaller with age. But this is not really the truth, because when we discuss with the experts worldwide, they're going to say, and this is something very crazy because this is not really published, that very often people which are looking at mitochondria from different tissue collect from aged organism, describe that sometimes they can find very big mitochondria, and they don't understand why. This process, which is here on the slide, can explain that. We can see that the mitochondria shape is done by different things, such as fusion and fission. What we know is that the fusion is altered, so that means that the fission is altered, and the fusion is still here, and the mitophagy is altered. So that means that finally, altogether, these different mechanisms may lead to have some bigger mitochondria. Okay? We don't know why, but if you analyze part by part, you can imagine that mitochondria should be bigger when we age. So, yeah?
So both responses are true?
No, both responses, I think it depends on different things. It depends on the tissue you are analyzing. But in my hand, for instance, I found in one muscle that the mitochondria were smaller in aging. So comparing mouse aging, 24 months old to 3 months old, it was smaller. But when we consider another muscle, another skeletal muscle, they are bigger. So sometimes you have some difficulty to understand. And finally, I would say that regarding the global literature we can see today, it seems that the size, how can I say that? The size is not important, but the size, besides the size, what is important is the structure, okay? But not really the size. Of course, some paper clearly show that fusion and fusion process are altered and mitophagy, etc. That could lead to alteration of the size of the mitochondria. Interestingly, this is something that is always observed. You know, and it has been presented by Hélène and Sophie. We know that within the mitochondria, the structure is totally altered. So they just present the alteration of crystia. So you can see that you are losing the crystia, the structuration, etc. This is crucial for the good functioning of the mitochondria. Okay, can you maybe put again this slide, Hélène or Sophie? You know, where you demonstrate that the crystia are totally lost. Yeah, you can see, hey, good crystia. You can see they are here, of course, and with CD, etc. You can see that you cannot find very good crystia. This is something I observe also. And also accumulation, of course, of vacuole, we already discussed about that. Well, altogether here, we can see that the structure of mitochondria, this is something that could be considered a marker of age. But what we don't know here, well, and what I'm going to maybe to say to you for the first time, because it's also an unpublished result, is that considering mitochondrial size is too difficult to consider as a good biomarker, because you will have a kind of Gaussian curve, you know, of the mitochondrial size and mitochondrial structure. So that means that if you measure the different mitochondria, you will have the size, or if you make a kind of index with the size and the structure, I don't know, there are a lot of different parameters, etc. Yeah, you will have this kind of thing. So that means that you don't know exactly, finally, if you consider, I don't know, tissue where you have more mitochondria that are in this part, or more mitochondria that are in this. So to try to fix this kind of thing, what we did in the lab, we did an analysis with artificial intelligence that consisted in analyzing all the mitochondria of a tissue by bioinformatics, okay? And we are able to say that finally we have different population of mitochondria. You have the small one, the big one, and the intermediary. And what is interesting is that this Gaussian curve is going to evaluate with age. So, and by tissues. You can find some tissues that are still respecting this kind of distribution, and some are going to lose this kind of distribution. So what is more important than size alone is the trajectory of the distribution of your mitochondria. So here again, we are not considering only size in this index, we are considering a lot of different markers, such as circularity, such as what we call the, I don't remember what is the name of that, but you know, the internal structural, but this is not the good word, but don't care. So we are compiling different features of mitochondria, putting together, just obtained by electronic microscopy. And by this way, we are able to predict the aging process of one individual from some mitochondria collected, okay? So this is something that in the future may be considered as a good biomarker of aging, not only the size, but the trajectory of the size, maybe, and other parameters, such as structural parameters, that are evaluating during aging. Here again, it's very difficult, as we discussed on telomere last Monday, it's very difficult to get an idea, a precise idea with a snapshot. The more easy is to consider a kind of trajectory. If your trajectory is still the same, that means that when you are young or when you are aged, you don't even change your mitochondrial shape. But what is important, if you consider that this shape is affected by a process during aging, you can measure that and you can say that, oh, sorry, but here, your patient, you can say to your patient, you are modifying your mitochondrial size or shape and dramatically, okay? So this is something that is important, and that can be also linked to the different things we discussed just before. Okay. First, the genetic. then the structure, and the third point that we can extract from the presentation of your colleague is the function. We never talk about the function. We talk about the Ross production, which is not the function. This is the consequence. What is the main function of the mitochondria? What is the main function? Production of ATP. What is the consequence of mitochondrial loss of function during aging? There is less ATP. So that means that in terms of energy supply, your skeletal muscles are not fueled correctly. Okay? How can we measure that? How can we measure that? If I want to consider that as a kind of biomarker.
Maybe
Tomorrow-
we can measure some enzymes which are related to the skeletal muscles, such as the CPK or whatever, although they are not really specific, but they can be associated with some muscle damage or lactate dehydrogenase.
Well, it's quite difficult to measure it directly. We use a technique which is kind of indirect technique because, you know, that will mean that to measure that directly the ATP, you need to have a kind of situation which is frozen situation, you know. You need to have exactly, it's something you can do, of course, in the lab, that is quite difficult to promote that clinically. So instead of measuring directly the ATP production, because it's very unstable, what do you propose? Oh, my question is, how do you measure the mitochondrial capacity?
Maybe by measuring the expression of some proteins and the respiratory chain, like it.
We can do that, but it's still very difficult, very easily, not easily, but very more simply. This is a consequence. This is something that we measure here at the different people, the strength of course, but this is not exactly what we mean. What would that mean? How can you measure the mitochondrial function? You want to measure the function of mitochondria. We said that mitochondria promote ATP production by using glucose and lipids. And so what can we measure when we want routinely measure the mitochondrial function? Come on.
The use of oxygen.
What is the name of that? The use of oxygen by mitochondria is, we are measuring the mitochondria. Yeah, respiration. Okay. So this is something that is done more and more. And this is something that you are measuring, for instance, when you do indirect calorimetry. Well, of course, it's more elaborate than just measuring the mitochondrial respiration, but this is totally linked. Okay. So if you have access to biopsy, or even if you have access to blood cell, you can consider, for instance, the mitochondrial respiration as a kind of good biomarker. And this is something that is done. Maybe some of you are directly concerned by this kind of thing. In the cohort here in Toulouse, which is called, you know, maybe this cohort, Inspire cohort. This is a big cohort of 1,000 people, aged from 30 to 100 years old. In this cohort, we collect blood cells, okay? We collect the PBMC, which are the mononuclear cells in the blood, sorry, and we measure the mitochondrial respiration. And we found that not only with chronic age, but with biological age, which is a different notion, taking into account the loss of intrinsic capacity of these people, we found that the mitochondrial respiration in the PBMC, so in the blood, is very correlated to the loss of function of the patients. So this is incredible, but this is something that is really promising, okay? So measuring mitochondrial respiration is crucial, maybe, to consider mitochondria as a good biomarker of aging. Why? Because respiration is something that deals with, of course, energy production, but also by the first thing we discussed together, by the growth production. If your respiration is increased in a context of aging, that means that your growth will be also increased, okay? So this is, of course, the first evidence showing that measuring respiration in mitochondrial respiration in aging will be considered as a good biomarker of aging. Aging, which is, for me here, in that case, progressive loss of intrinsic capacity. Okay. So they finished their presentation by, yeah.
Je crois que c'est à la dernière diapo. Du coup, j'ai compris qu'il préconisait de faire une restriction calorique et l'activité, enfin, la pratique d'une activité physique.
Yeah.
Mais ça, c'est préventionnel ou même quand c'est installé, parce que du coup, vu qu'on sait que la biogénèse mitochondriale, elle diminue chez les personnes âgées, du coup, il y a moins d'ATP, donc les muscles, ils en ont moins, donc il y a moins de régénération pour les muscles qui en demandent beaucoup. Et en plus, on fait une restriction calorique et du coup, il y a moins de synthèse protéique.
Alors, je traduis d'abord et après on répond. So Leah asked a question about the caloric restriction and wants to know if finally caloric restriction or physical exercise should be, so this is something that should be done as a preventive strategy, or even if it, the loss of function is already there, can we maybe say that it's difficult to recover some mitochondrial function when the alteration is already there? Hélène and Sophie, maybe you want to say a word or not at all about that, about these two different things, physical activity and restriction calorie. This is a transition to the next publication that will be presented just in 1 minute. Do you have some words to say about that? Maybe in French, if you want to answer to Léa. What do we know about that?
De restriction, la restriction calorique, en gros, ça permet, c'est qu'il y a moins de dommages à l'ADN mitochondrial. Et du coup, ça fait que ça peut permettre de rallonger. Pour le vieillissement, c'est du coup meilleur.
We are going to have a presentation about caloric restriction, so we are going to know maybe a little bit more. This is exactly the same for physical activity. We have to consider mitochondria as a kind of, very important power plant center, kind of power plant, receiving some energy substrates such as glucose and lipids and whatever, and transforming this kind of substrate to ATP, so the major source of energy, of course, of your organism. Well, by promoting restrictive caloric restriction, what you are doing is just that you are trying to maybe decrease the level of substrate that is available for your mitochondria. Okay, by this way, you are going to slow down the process of growth production. That's it, finally. Okay. In the other hand, when you do physical activity, this is totally different. Not totally different, but it's quite a bit different. First of all, of course, you are going to use better your substrate. Okay, so that means that less storage, and I'm not going to come back to your metabolic courses, but that if you use more, you store less, and then the storing is something that is not really good for your health, and particularly for the metabolism aspect. Okay. Well, when you promote and when you do physical activity, you should know that you are going to increase the number through the PGC1 alpha pathway and PPAR gamma. You are going to increase the number of mitochondria. By increasing the number of mitochondria and their efficiency, you are going to promote a good utilization of the substrate by the different mitochondria and the less production of growth. Okay, so taking together, you can see that these different approaches, and of course there are also other benefits. Maybe we are going to discuss with the next group, which is going to present the caloric restriction. But this is something crucial to understand that yes, we can act on our mitochondria, we can act differently, and this is something really important in the field of considering mitochondria as maybe a future biotherapy. This is not the topic of today, but based on this kind of observation and based on what we just discussed before, some people, and we are these people here in Toulouse, consider mitochondria as maybe a good future biotherapy. You know what is biotherapy? It's therapy done from a biosystem. So what we are able to do is to collect the mitochondria, select the good and the bad one, and re-inject only the good one to our organism, fish, for instance. And by this way, we are able to say that mitotherapy is something that we can do in the future. This is not something that we created. mitotherapy exists in your body. For instance, I have in my mind a publication done in 2015 showing that if you disrupt the bronchoalveolar epithelium, you can disrupt that. This is something that I done during my postdoc by different chemical techniques. You have a description of your bronchoalveolar epithelia. Some cells that are immune cells are going to arrive on this epithelia, and by making a kind of bridge, which is a connecting 5 bridge, they are going to exchange the mitochondria. Okay, so that's quite important to think that and to know that mitochondrial blood therapy, this is something that finally already exists naturally in your body, and we can do that, we could do that in the future as maybe a therapy for treating age-related pathways. Okay, here again, another thing, last one, we never talk about that before switching to the other presentation, but mitochondria can be retrieved in the blood, not in blood cells. Really like that in blood. So that means that just by a blood withdrawal, you should be able also to have maybe an idea about mitochondria size, function, DNA, etc. Okay? We didn't talk about that because it's not something that is really current in the literature. Yes? Last one.
Moi, j'ai une question. Vous avez dit que du coup, la théorie de la mitochondrie, c'est basé sur une balance où on avait une production de rose et une production d'antioxydants, et qu'avec le temps, avec là, du coup, soit on avait une production excessive de rose, soit
les systèmes antioxydants étaient.
Est-ce qu'il y a des études où, par exemple, ils ont complémenté les gens avec des antioxydants? Est-ce que ça a marché?
Oui. All the studies regarding the use of antioxidant as a pharmacology of mitochondrial theory of aging works. But unfortunately, that is unusual. The imbalance at one point is going to be too big to be compensated by the use of this antioxidant. The use of, when you say that we are going to use antioxidant therapy, this is not directly antioxidant that you give, okay? You are giving some elements that are going to potentialize your antioxidant enzymes, such as SOD or catalase. Okay, you are going to give to your patient some zinc, some potassium, so etc., etc. But finally, you are just helping this vitamin, the enzyme. You are not giving the enzyme. And you cannot boost the enzyme more than they are able to do. Okay? So at one point, we know that, of course, antioxidant strategies are not working anymore because, of course, the imbalance is too high. But this is a good point. And I mean that scientifically, acting through oxidation and trying to decrease the rose production is something that could work. Okay. We have to switch to the other publication because we don't have too much time today, especially. Can we have the group of caloric restriction first, and we will finish with the mutation? We don't have a specific order. So just for maybe for the people that are online, Yamina proposed respiratory cycle, and morphological change is something we discussed. We're doing it, and Zaina, we measure enzyme associated with muscle damage, stuff like that. And Marianne proposed oxygen consumption rate. This is exactly what we thought about respiration. Do you have a memory stick? Put it here and the folder. People that are in line, I would love you to switch off your camera if you are not on the lab, or if you that's something that would be more interesting for us to see you when we discuss. No. Okay. So Alexandre, Borian, and Morgan are going to present the second one, which is dedicated to the finally the absolute proof of concept that mitochondrial metabolism is totally linked with age. So the evidence, the absolute evidence, is given by the fact that if you, how can I say that? If you decrease the load that is on metabolism and mitochondria, you are going to live more long, but maybe also better. Let's see. Okay, the floor is yours.
Okay, so hello everyone, I'm going to present this paper.
Maybe if you put it full screen on the low right corner, you see tu vois l'icône avec la page. Si tu appuies dessus, tu peux te mettre en plein écran. Voilà. En bas, plein écran.
So with aging, we know the body know many physical changes. The most one is sarcopenia. So sarcopenia is a progressive loss of muscle mass and strength. This is affecting mobility, quality of life, and also can increase the risk of falls for aging people. Skeletal muscle are divided into two main types. So first we have oxidative muscle with slow fiber, so fiber type 1. They could be of endurance activity. And then we are a lipolytic muscle with fast fiber type 2, which is for intense efforts. They are not affected equally by the aging. Lipolytic muscle are most affected by the aging. And moreover, mitochondrial morphology and dynamics vary between oxidative and glycolytic fiber, so influencing their ability to produce energy according to the intensity and the duration of the effort. For the study, the author used rats, and they are divided into 3 groups, compared for the effects of the aging alone and the aging associated with caloric restriction. So the first group is composed by young rats that fed ad libitum with free access to food, and there are 4 in this group. The second group is composed by old rats and then feed ad libitum too, there are 4 also. And the third group is composed by old rats and they are in caloric restriction during 30 months. And there are 11 in this group. So the caloric restriction in this paper, they are focused on the caloric restriction, and they are take support on the paper of Gilles Goupillou. And this author says caloric restriction is good for health in aging. This technique consists in gradually reducing food intake to around 60 to 70%, will maintain essential nutrients and avoid malnutrition. And the aim of this study was to determine the impact of the caloric restriction compared with a conventional diet on the muscle fiber composition and mitochondrial morphology in aging muscle.
So the first result is the effect of aging and caloric restriction on body weight and muscle weight. So in the graph, we can see 5 type of muscles and the muscles to body weight. We can see all the group with a normal diet have less muscle to body weight than adult groups, normal diet, and aging groups caloric restriction. And its significance in tibia anterior, gastrocnemius, and pancreas muscles. So we can conclude caloric restriction prevents the aging relate, loss of relative muscle mass. Okay. So the second result is the effect of aging and caloric restriction on fiber cross-sectional area and fiber type. So on the left, we have the proportion of type 1 and type 2A fiber type of muscle. We can see aging groups with normal diet have more type 1 and less type 2A fiber muscle than adult groups and normal diet, and aging groups calorie restriction. And on the right, we have the same result. In also on the left is a soleus muscle, is an oxidative muscle. On the right is the gastrocnemius muscle, and it's glycolytic muscles. So we have the same result for the type 1 and type 2A, and we have for the groups aging with normal diets, less type 2B fiber type muscle and more type 2X fiber type muscle than adult groups and aging groups with caloric restriction. So we can conclude caloric restriction prevents the effect of aging on muscle fiber type composition.
So here they wanted to see the effects on of the aging on skeletal muscle mitochondrial content. So in the first graph, they evaluated the mitochondrial volume density, and we can see that there is no difference between the three conditions for every type of muscle. So aging and caloric restriction don't have any effect on the mitochondrial content in the skeletal muscle. So in the second and third graph, they wanted to look at the mitochondrial activity in the two types of muscle. They use the CDH stain to follow this activity. SDH is a succinate heterogeneous, which is part of the complex for the respiratory chain. We can see that in the soul, we have a decrease of this activity with aging, and the caloric restriction doesn't have any effect. But in the gas, we see a decrease of the SDH activity with aging, and we can see an increase of this activity with the caloric restriction compared to the aging without caloric restriction. So we can say that caloric restriction did not protect against the effect of aging on the CDH activity in the soul, suggesting that not all muscles are protected from aging induced decline in mitochondrial function by caloric restriction. So finally, they wanted to look at the mitochondrial dynamics. So they took three proteins, MFN2, which is a pre-fusion protein, and the RP1 and FIS1, which are pro-fission protein. They measure the protein content in each muscle, also in the gas. For the soul, we can see an increase of DRP1 with aging, and in the caloric restriction condition, we can see an increase of MFN2 and DRP1. So in the soul, we can say that aging seems to increase mitochondrial function. For the gas, we can see an increase of every protein with aging, and the caloric restriction seems to partially attenuate this increase. So their hypothesis was that an increase in the expression of both pro-fusion and pro-fission protein might reflect an attempt to compensate for an aging-related increase in mitochondrial dysfunction. So by first increasing the fusion to meet the matrix content in healthy mitochondria, with that of dysfunctionality in order to reduce mitochondrial dysfunction, and in the second time increase fission to segregate the most dysfunctional mitochondria to degrade them via autophagy.
To conclude, caloric restriction appears to be an effective protection against the effect of aging. It helps to preserve muscle fiber composition and mitochondrial function, in particular in muscle with high oxidative activity. And in the future, it could be interesting to study with human, and for strategy,
for delaying sarcopenia and maintain muscle health as we age.
Me? Me? Okay. What is your opinion about the question raised in the audience? You could have a question because it's an easy publication. So imagine that you have one speaker that presents his result and you want to discuss with him or with her. You should have question. You are student, you have question. Yeah.
It's a very simple question. Why did they use so less mice or rats? Because there were 4, 4, and 11. So I don't know if it's pretty.
That's more a remark than a question, but yeah, we can of course note that the number of rats was very low, and that's why maybe also there are a lot of I can tell that the distribution is quite wide sometimes. So I think that if you want to conclude as they did, they should increase the number of animals, and if you are a reviewer, you ask them to increase the number of rats here. Other publication also this demonstrate exactly the same thing in other more prestigious journals. So, but this is not a question. I mean, maybe if you want to complete, but no. Other question, other remark? Okay, first of all, number of rats is not enough. Yeah.
Another remark is they didn't use young rats in caloric restriction, only old, so they would have maybe seen a better response, because of aging fibers.
Yeah. But this is criticism here. Do you have something to say about that? They don't use young rats, particularly under caloric restriction. You have an idea why? Not why, but can you say a word like that? Because it's not really a question. Again, this is a commentary. What do you think about that? Is it something that should be very informative to have young rats under caloric restriction? Regarding the question they raised? What do you think? Just discuss. It's not there is no precise answer. What do you think about that? In English or in French, if you want to switch.
I guess.
Yeah, you think? I mean that she's right, absolutely. But if you reconsider the question raised by the authors, they just want to know what are the effects of caloric restriction during aging. They don't want to know what's happened when we are young and under caloric restriction. The question she raised was about the fact finally, can we? It's something that we can say. Can we consider that the results they get are specific of age, or we can retrieve exactly the same with young mice? So that's something we can ask as a question. But in that field, if you want to know if caloric restriction is something interesting for age animals or for age individuals, they don't need to get some control. They put the control, which is a young rat without ad libitum. But that's it. That's enough to compare. This is something I did also. You know, I was confronted with this kind of question in one paper when we treat the mice with a drug which is called, not a drug, but a peptide, which is called apelin. I had three groups: 3 months old, 12 months old, and 24 months old mice. In reality, I treat the 3 groups. So we have controlled PPS and treated in the 3 groups. And I removed the 3 months old. Why? Just because in one experiment, and I had something like more than 120 feature, different things that we measure. On one parameter, I found that the treatment induced an increase of muscle function. And finally, one of the reviewers told me, but your molecule is an anabolic molecule. So that means that maybe it can be considered as not an anti-aging drug, but an anabolic molecule, such as the anabolic you can use for when you do some muscle fitness. And finally, he considered me that you should remove the group, which is 3 months and treated, to get the idea that. So here we cannot exclude that maybe they did also this group. They have an effect, and they don't want to present because of the message of the publication. Other comment, other question, other remark, yeah?
why the caloric restriction is effective on really some muscle. And my second question is if calorie restriction is bad for some muscle in aging.
So first of all, why can you give us your opinion about some muscles that are sensitive, that the other that are not? Quel est ton point de vue? Les muscles qui sont sensibles et d'autres qui ne le sont pas vis-à-vis de la calorie restriction. Vous avez... Je crois que c'était toi, oui, Alexandre, qui avait un truc là-dessus.
Mais
In French or in English? If you want in French, I will translate. Prefer in English, you do in English.
je n'ai pas compris parce qu'ils sont plus sensibles.
So they are all sensitive. Why? No, because it's not maybe Alexandre. It's I don't know the name. It's this one. You can see that in the storage we don't have any effect about calorie restriction on the succinate enzyme, rather than in the gastrocnemius we have an effect. What is your point of view? What is your hypothesis here? And what is the hypothesis raised by the author? You start an explanation, I think. Yes? No? Yeah, you can continue.
Du coup, pour moi, c'était que ils disent que les fibres glycolytiques, elles sont plus affectées par le vieillissement. Et pour moi, ce serait pour ça qu'elles sont plus sensibles à la calorie.
Well, the glycolytic muscles are more affected by aging, and this is something that is well known. And what we know, if you consider fiber, we don't know why, but there is a switch. This is something that has been presented by Alexandra anyway. There is a switch between type 2, whatever the type 2, B, X, DA, and type 1. So you switch during aging from glycolytic fibers to oxidative fibers. That's very surprising. That's very surprising. Because we just mentioned that mitochondria is defective, is altered during aging. And you switch from fiber that are more glycolytic to more oxidative. There is an explanation for that. This is not the topic of today. We should make a workshop dedicated to sarcopenia and muscle. One of the explanations, one of the possible explanations, is that we are not looking at the same thing. When you are young, you have, I don't know, 1 million fiber per muscle. Let's imagine 1 million fiber per muscle. Okay, when you are young. When you age, you are losing some fiber. So you have only, I don't know, 500,000 fiber per muscle. And you are losing more glycolytic than oxidative fiber. We don't know why, but we don't know why. We know a little bit about innervation of this fiber, which is more prone to be altered of the glycolytic fiber than the oxidative one. Okay. So when you are losing the innervation of this fiber, you are also losing the fiber. Okay. So what we know is that it's a proportion finally, and here you can see that's proportions. Okay. So in proportion, we don't know exactly why, we cannot explain, but we often find this kind of result. There is an increase, there is a decrease of glycolytic fiber, and there is an increase of type 1 fiber. But finally, this is just proportion. So one of the explanations could reside, of course, in the fact that we are measuring different muscles, and these different muscles are not how can I say that, composed by the same fiber. Okay. Other question, other remark? Yeah, again.
Can you explain the difference between the different types of two fibers?
Do you know that? Did you check?
Type 1 is oxidative fiber.
Okay.
Fiber, and
commonly, we say that commonly they are the red fiber. They are more, the muscle are more red. Okay.
And type 2 is the glycolytic fiber.
Okay, glycolytic. And do you know did you hear something about the fast and the slow fiber?
Yes, type 1 is slow.
Yeah.
2 is fast.
Okay. So if I resume what we said, during aging, there are more type 1 than type 2. Okay? More type 1 than type 2. So that means that there are more oxidative than glycolytic. This is totally counterintuitive with the fact that mitochondria is altered. Why after that? Why after that it's a little bit counterintuitive to say that During aging, there are more oxidative fiber than glycolytic. What do we know about metabolism of these fibers?
Are less effective. So
No, that's why, yeah, but what is the reason why I say that? Yeah? Because in oxidative fiber, you have more mitochondria than in glycolytic. And we just announced before that there are less mitochondria, they are less prone to, etc. So we don't know exactly, according to the different experts, there is a relationship between innervation of glycolytic fiber and loss of this portfolio of this type of fiber in profit, of course, of oxidative fiber. Okay. Okay. Other question, other comments about this presentation? We are talking about caloric restriction. Are you interested in caloric restriction or no? Is it something that you can consider as a kind of therapeutic or strategy to fight against aging? What is the big side effect of this strategy? Which is really important, and of course that they didn't talk about here. We are talking about rats, we are not rats. What's happening if you promote caloric restriction in humans? But just before starting, and then what is caloric restriction in human? What is your opinion? What is exactly the process?
It's to be lower than the metabolic needed.
Yeah. So according to you, how many percent are you decreasing the calorie intake?
I would say 20–30%.
Yeah. Okay. This is around 30%. Okay. First of all, 30% is quite difficult, and if you are aged, it's more difficult. Okay. So first, then what is the main consequence of that?
I would say mental difficulties, maybe.
Yeah, we can have some difficulty to try to follow this protocol, and of course sometimes you have some abandon, but it's not the main result. If you consider that your patient are following the protocol,
if there is not enough protein intake, we can lose muscle.
that's the main result of all the caloric restriction protocol provided in humans. The first thing that moves is the skeletal muscle mass. And very surprisingly, compared to this kind of paper, we are losing muscle mass. Before losing fat mass, we are losing muscle mass. That's also true in the field of obesity. Field of obesity, if you eat less, you are going to lose muscle. When you are aged, this is dramatic because this muscle you are not going to recover. Okay, that's the main issue, for instance, of people right now that are promoting some medicine. You should hear about that in the media. You know, the Vegowi medicine or some stuff like that. The anti-obesity medicine. But what we don't know about this GLP-1 agonist is that they are promoting, first of all, because of low food intake, they are promoting a loss of muscle mass. When you are aged, this is dramatic. Okay, that's why if you want to associate this protocol to age people, you have also to associate other compensatory stuff, such as protein intake, maybe. But here again, you cannot propose an intake that is more important than the capacity of the individual, and other products that are able to stabilize or at least to maintain the muscle mass. Okay, this is not the topic of today. Okay, caloric restriction is something that is, as you mentioned, very difficult to follow. What are the other alternatives, which are really well studied right now, but directly connected with this kind of experiments? You present something about caloric restriction and the consequence of on aging. We can also talk about, all these experiments done on C. elegans, or in flies or whatever, in different species, showing that caloric restriction is going to increase lifespan about 30%. This is something that has been done 20 years ago, and this is not something that is we are going to discuss today. But regarding the strategy, caloric restriction, what do you think? You just mentioned that it's difficult. What are the alternatives? Yeah.
Intermittent fasting.
We can promote also the intermittent fasting. You know what it is?
Mm-hmm.
Okay, 24-hour with food, 24-hour fasting. Okay, here again, very difficult to follow. very difficult, psychologically very difficult. What is the third one? You hear about that, I'm sure. It's a West Coast team, which is the name of the last author is Panda. And this is a French student that did this very interesting first observation in mice, and now they are going to do that in humans. What we are talking about. Not calorie restriction, not intermittent fasting, but the last one that consider also the biological clock. What it is? What is the name? You never heard about the time restricted feeding? You know these people that eat only
Like
from
keto diet?
yeah. Yes, that's it. These people that only eat from noon to 8 p.m. So it's 8:70, 16, sorry, 8:16. During 8 hours they eat, and during 16 hours, consecutive 16 hours, they fast. Okay, so basically it starts at noon, okay, 12, until 8 p.m. After 8 p.m., you don't have the right to eat. Before noon, you don't have the right to eat. You just can drink. By this way, we know that compared to calorie restriction, you should have very interesting effect. You don't eat more, apparently. You don't eat less. That's interesting also. In terms of caloric intake, this is exactly the same. But because of chronological biology and clock, you have apparently a better way to use your substrate, your energetic substrate. Okay, and it decreases considerably all the aging processes and all the age-related pathology. Yes.
On Panda diet, the person can eat until 16 or 8 p.m.
No, they have to wait from noon to 8 p.m. Okay. I'm talking about the individuals, humans.
Yes.
Not on the mice. The mice is quite different because it's difficult. The mice are, inverted cycle. So if I remember when we did that here in our hand, we did. I think that we start the mice from 7 p.m. because they are eating during the night, and the morning after we put the animals. Okay. But in human, this is something that is quite observed that starting eating at noon and finishing at 8 p.m. Okay, you can of course say that I prefer to do 9:15. 9:15 is you start to eat at noon or 11, but it's quite important to try to be regular and to preserve a kind of rhythm like that. And apparently, it changes everything. Okay. So at least on the current aging process, it decreases the aging process. Okay.
Speaking about this kind of treatment, this calorie protocols, I think that it's not easy for the aged person.
Well,
because
I know some of them that did that.
Yes, but when we talk about each person with other pathologies and like diabetes, and they can't do this.
I know one people at least, which was he was not obese, he was overweight, and he did that. He was a researcher, a very famous one. I can't say the name because no retreat. His name is very well known people here in Toulouse in the field of metabolism and obesity. It's Max Lafontaine. And this researcher, one day at the institute, I saw him, and he was a little overweight. And I said, "Oh, Max, I have the feeling that you are better." He said, "Cedric, listen to me. I start something that is very important and very interesting. I start the TRF. I said, what it is exactly? It was something like 5 or 6 years ago. And I start to eat at noon, and I finish at 8. And it changed all my life. I had some blood glucose, elevated blood glucose, it's finished. And I hit this guy was a good eater, and he said, I did exactly the same thing than before, but only during this. I don't know where it is. Maybe I hope it's not that, but no, I know it's not that. But I don't know, if you can do that in a long-term period. But I think that of course our organism is not planned to eat, to be alimented, to be fed by different substrates all over the day. some people that are eating very late during the night, or, we know exactly that if you do that all along your life, you're going to have, of course, bad consequences, not only on metabolism, but even on aging process. You cannot imagine how the circadian rhythm are quite important and crucial for aging process. For instance, we know that the pilot of plane, the pilot that are, and not only the pilot, all the personnel that are working in the plane, they are aging quite rapidly compared to the general population. Why? Because of the jet lag. If you are always jet lagged, you will have some process in your body that accelerated aging. Okay. Based on that, some people in US developed a model of mouse they call jet lag. What did they do? Usually, I don't know if you already went to the animal facility, but the lights are switching off at 8 p.m. and switching on at 8 a.m. Okay, this is something that is really regular and automatic. If you delay each day of 5 to 10 minutes, the switch off and the switch on, the mice are going to age rapidly compared to the control one. That's something very surprising. So the respect of rhythm are crucial. And of course, in our society, because of the food abundance and all the stimulation we can have with the screen, we are going to age. It will be very difficult to age healthy in the future because of this different thing. So that's one. It's quite important to analyze the consequence of such thing and possibly on TRF on aging process, I think. Other question, other remark? Yeah?
Does the citrus diet can be also an alternative?
Which one?
The citrus diet.
Citrus diet? I don't know that, what it is. Really? Why? Because maybe of phenol composition or polyphenol stuff, or?
I think so.
Yes.
That it can
Yeah, maybe. You have a lot of things like that, that are published in different things. A few years ago, I had a module in M1, which was dedicated to nutrition and different topics. And I had one which was dedicated to nutrition and aging. You know what is the French paradox? You know what it is? In the Southwest particularly. In the Southwest, we are eating ducks. You know what it is? What? Okay, we are eating ducks. And we have a kind of survival curve which is higher than the other part in France, and even if we compare France to other countries. Okay? And why? We say that it's a French paradox because in the Southwest we are eating the fat of this duck, which is really rich in polyunsaturated fatty acids, and also we are drinking wine, red wine, and altogether these two things are going to bring you a lot of positive things, such as polyphenol in the wine and polyunsaturated lipids. So alimentation is crucial, of course, but it's not the only thing, but alimentation is crucial. Here we just bring the proof of concept that because we have to come back on mitochondria, if we decrease the load on mitochondria, you are going to increase your lifespan and probably your health span. Okay, so that means that mitochondria is crucial in the field of aging. Considering mitochondria as a biomarker, we discussed just before, is also important in the future. And this kind of experiment should lead, of course, to maybe some positive and negative control to see if the biomarker can be validated on. Other question, other comment? We can discuss during hours and hours for that, but we don't have the time to detail. The last one is about the mutation. So thank you very much from the group. What is supposed to represent the mutation? Maybe online. I have the name. Yes. Last Monday. Will be more significant to OCR. It was okay. Last Monday, some of you asked me about the presentation. Of course, what we are going to do, I'm going to collect on the presentation. collect all the presentation and do share with you on the presentation through Moodle or through the Google Drive. This is not the reason for stopping to take some notes. You don't need to write what you're going to say because this is something that is spontaneous. This one. When you read, it's less intensive, finally. And I'm sure that you know that's what you're going to say, but do it by your own. Do whatever you want. Okay, we go. We're going to listen Marie, Manon, and Rama about this paper regarding the germline mitochondrial DNA. You know, we talked about the DNA mutation, particularly in the mitochondria, aggravate aging and can exert brain development. Here we go.
Aging results from a progressive accumulation of cellular damage will lead to the aging. This dysfunction I linked to sequential variants in the mitochondrial DNA sequence. And the key question is to what extent hereditary mutation in this mitochondrial DNA contribute to the aging process. A recent study reveals that mitochondrial DNA mutation passed down from the mother have a notable effect. This appears to predict reduced fertility in heterozygous mouse, carrying to the mitochondrial DNA premature aging. This finding suggests that the maternal mitochondrial DNA mutation could similarly exert some aspect of normal aging in humans. This study explores the hypothesis that the germline mitochondrial DNA mutation not only worsens aging-related phenotype, but may also have a detrimental effect on brain development. The study aim is to investigate the impact of maternity-inherited mitochondrial DNA mutation on aging, assess how this mutation affects brain development, and determine whether mitochondrial DNA mutation aggravates premature aging and phenotype. The researchers generate a series of mutant mice to study how mitochondrial DNA mutation affect aging. And for that, they use a poly-A wild-type mute mice passed on a very few mitochondrial DNA. Female, and the cross with a male Colgate white type and mutation for generate three first type of mice. The first one is white type. The second type is edge Colgate white type with mutation, and the third one is the mice with mutation homozygous. So then the researcher crossed male Colgate white type mutation mice with a white type female to produce the type 4 normal mice without maternal.
For those that are online.
Sorry.
Yeah.
The mice with mutation and no white type, sorry. And the mice with the type 5 mice with mutation and white type. So then they use male with heterozygous with mutation and cross with female with mutation, with white type, and the other one is knocked down for called A. So the type 6 is mice with tau and mutation.
Can you interrupt?
Yes.
Yeah. Can you explain, if you have it, what was the question raised before doing that? You know, why they did the first one, why they did we can understand that But what was the question? Why Paul A, one Paul, what is it exactly for people that didn't read the paper? Why they muted Paul, why they mute the Paul A, etc., etc.
They want to know if it's real if it's really from the mother mutation, and they do different type, Y type, so they don't have mutation, and the Y type with mutation is when the dad gives the mutation, and the other one is when the mother gives mutation. It's for confirm the big impact on aging and on brain development.
Yeah, but why they did this hypothesis that by mutating only the male, only the female in the couple, it should affect differently the pups? Why they did that? What is the first hypothesis? Pourquoi ils mutent que le père ou que la mère, ou les deux? C'est quoi l'hypothèse de départ? Est-ce que vous l'avez vu?
Que c'est l'ADN mitochondrial qui vient de la mère.
Okay, because we know that mitochondrial DNA comes from mother. If you are mutating only the father, of course, normally you know have some effect. But if you mutate the mother, you will have the inheritance of the mother DNA mutation on the mitochondrial DNA. That was the starting hypothesis. Sorry for you. Continue.
So to confirm the hypothesis, the researchers analyzed the impact of mtDNA mutations on aging, reproduction, and brain development. There was a progressive decline in litter size, in the number of litters, and in the proportion of viable POLQ-A mutant mice across generations, from generation 1 to generation 4. The reintroduction of wild-type mtDNA slowed this negative trend, contrasting with POLQ-A wild-type mute process, which continued to decline steadily. This indicates that mtDNA mutations compromise fertility and offspring survival over generations. However, the reintroduction of wild-type mtDNA can partially mitigate this adverse effect. To investigate the longer-term impact of these congenerational effects on fertility and survival, the researchers then analyzed how the mutation influenced signs of premature aging. They observed increases in body weight and phenotypic aging score with successive generation. The POLQ-A mute mice showed a significant reduction in longevity compared to wild-type mice, along with an elevated Earth-to-body weight ratio and a decreased respiratory chain activity in tissue samples. These findings show that accumulating mtDNA mutations accelerates aging in mice, producing early signs of physical aging and a reduction in overall survival. Finally, the researchers examined whether these mtDNA mutations influence brain and neurological development. They observed structural abnormalities in the brain of mice with mtDNA mutations, including disruptions in the striatum and altered hippocampal shape. There was noticeable alteration in cortical structure and hippocampal lamination, along with abnormal distribution of NER1, a neural marker in mutant mice. The mtDNA mutations disrupt brain development and organization, particularly in areas critical to cognitive and motor functions.
To conclude, we know that the mitochondria are involved in several cellular functions. And in this article, they show us that the mutation
in mitochondrial DNA inherited from the mother decrease the fertility, the tissue function, and the lifespan. And in addition, they prove that the mitochondrial DNA mutation inherited by the mother combined with the somatic mutation accumulated over several generations, increase aging phenotype and brain malformation. And this result suggests that the mitochondrial defect caused by is caused human neurodevelopmental disorder and the brain aging process. The limit of this article is maybe the model. They use mouse. Maybe it's not the best model, and they can reproduce their results in another model. And they don't study the effect of the environment on the dysfunction of the brain. And to the perspective of this article, opening potential avenues for therapeutic intervention targeting mitochondrial function, prevent aging-related and neurodegenerative disease. Maybe they can use CRISPR-Cas9 to delay the mutation or develop the treatment antioxidative stress.
Thank you very much. Here we go. Question, remark, commentary. Don't raise your hand. Take the mic directly.
So it's a question about the different regions. Which model can you choose to reproduce mitochondrial disease if the mouse model is not good? You have an idea?
When you announce this kind of thing, you have to be ready for the question. And the model is not good, and then we should do other models. You should have the question. I have also the same question. First of all, why the model is not very good for you? You critique you criticize the model, but you don't say why. Why mouse model is not good?
In other articles, we know they use other model like Cedric to study mitochondrial dysfunction.
Yeah.
But here to study brain and
so which other one, which model you prefer to do that on Cedric than on mice? This is what you mean.
For the brain development, I think it's better to use cat model, but it's not easy. It's really not easy and it's very expensive.
And for doing this kind of experiment, remember that you promote some mutation, blah blah blah, with the cross, etc. I think that is quite difficult. So using mouse is not a mouse are not good model for aging processes. Definitely, because you have to wait at least 2 years, etc. This is the lifespan of mice is quite long. Considering other models such as the C. elegans is quite interesting because the lifespan is something like, if I remember, 30 days. It's quite true also for Drosophila, so the flies. It's even something like 45 days, the lifespan. And you should know, maybe, I don't know if we talked about that before, but today we don't have the time again. We developed in my institute a new model, not a new model, but a very interesting model of aging, which is a fish. The name is Notobranchus furseri. For the basic name is African killifish, because this guy is living, in Africa and precisely in Mozambique, in Zimbabwe. And very interestingly, this model resumed all the hallmark of aging that we presented on Monday, the paper done by Lopez, in something like 4 to 5 months. I don't have the time to explain why this is a shorter lifespan, but this is a vertebrae that are displaying the shorter lifespan done among the different data, okay? First of all, the models, okay, maybe we can choose the models, but it's quite difficult for this kind of experiment. Other questions? Yeah.
Malformations qu'on observe au niveau du cerveau, là, comme elles sont dans certaines zones et pas dans d'autres, parce que je me dis, vous savez pourquoi? C'est parce qu'ils sont plus sensibles, plus énergivores, j'en sais rien.
Why we can observe some brain malformation only different area and not in other ones? Do you have an explanation for that?
Maybe because I know that have stem cell, and because have we say reproduction of cell, it can maybe use more mitochondria for that than the other part of the brain. I don't know exactly.
You said that the mitochondria, you said, sorry, that hippocampal
Yes.
has more stem cells than other area?
Yes, he have a little bit of, he have a part.
Hmm? And this is the explanation done by the author, not alone.
No, not by,
That's true.
just because I know so.
Yeah, and you know, and they explain you that the hippocampus is combined with more stem cells. Okay, this is something that I didn't know. I'm not an expert of hippocampus, of course, but I have some stuff to on that, and I don't have the feeling that there are a lot of that. But maybe there could be an explanation. What should be the other explanation? Maybe that is directly connected with what you said. I think that the step agreeing with the number of stem cells, if it's not something that is sure, but you can just switch saying that according to me, we can exercise that the area that are more targeted by the mutation are containing more mitochondria, of course. So, but I don't know if it's the case or not. But the only explanation that we can have is reside in this hypothesis. The question of the comment, yeah, I'm sure you have, yeah.
That's in the article they say that the mutation of the mitochondrial DNA mutation of somatic DNA.
Can you repeat this?
And just to check, they say that the increase of mitochondrial DNA mutation increases the rate of mutation on DNA somatic.
Mm-hmm.
Is it that they say?
Yes. I mean that here, all the publication reside in the fact that the mother is going to transmit the mutation. So this is a germline that is affected. Is there a question?
No.
Oh, sorry. I answer to another question. I mean, what is the question?
It's only germinal.
In that case, they admit that by targeting the mitochondria in mother or in father, you are going to, of course, target the germline, and through the germline transmission, you will have, of course, a mutation that is also in the newborn. But it was not a question. We are talking about inheritance, which is something that is not. Yeah, it's okay. It's okay. We are here for discussion, so don't worry for that. Other question, other comment? What do you think about this paper? Yeah. You have a question.
Say, like, do we know famous mutation in mitochondrial DNA that can lead to neurological aging diseases, and can we use them as biomarkers of brain aging?
That's a good question. Maybe Hélène and Sophie also can answer because they identify some precise mutation. So the question right here is, do we know about precise mutation that can induce maybe something particularly in the brain? Apparently here you don't have the answer. I don't have also, so I don't know, but I think so. I think that we lost Hélène and Sophie. Next time, I'm going to ask you to switch on your camera because people that are here are more respecting you than you are doing. So this is not serious. But to try to maybe to complete the question or to go forward on the question, I think that we know that not one, but a kind of couple of mutation can be associated with, not directly maybe with the tissues, because I'm not sure, but I read something about the fact that some kind of mutation may be affected to a more dramatic consequence than the other one. So we know that a combination of mutation can just, but this is the starting point, you know, how understanding finally the consequence of mitochondrial DNA mutation on aging process. So we are just knowing that we have association of mutation that could be dedicated to not directly to loss of cognition or of motility, but at least to a tissue or function. For the question, for the remark, for the thing, what do you think about this paper? What do you think? This is complicated because it's a little bit of genetics, but at the end, the take-home message is quite clear, saying that if we mutated the mitochondrial DNA, we have some acceleration of aging process. What is lacking for you in this publication? Something crucial. What is missing in the publication? You maybe. You have any critics on the paper? Not at all. It's perfect. No, it's not perfect. First of all, you are promoting mutation. Then you are measuring the effect of the mutation on, I don't know, on the reproduction, on etc. What is the missing step between? You are convinced, that's okay. Did they check the mutation of the pups? I'm not sure. I'm not sure they did. They are convinced that if you are mutating the male and the female, and if you are genotyping them, if they have the mutation or no, but that doesn't mean that they are sequencing the DNA, the mitochondrial DNA. So they don't do that. So that means that we cannot confirm that the precise mutation to answer to the question is here. We know that the pole A is mutated. That doesn't mean that the mitochondrial DNA will be mutated on the good way we want. What is the answer for that? What should be promoted to do that? You want to mimic what have been raised by Hélène and Sophie in the first presentation. You want to induce some specific mutation. What do you do? You want to do that. You don't want to measure, you want to do that. You want to, I want this mice to present, I don't know, the mutation of arginine 162. What I have to do? How does it come? Yeah, you have to do transfection to make what they call directed mutagenesis. Okay, you are going to make some directed mutagenesis and punctual mutation. This is not what they've done. They are doing something more larger and broad. You are mutating one of the enzymes that is able to make of course, the replication of the DNA. Yes?
Maybe I understood, but what does all GA gene does in the daily
The polymerase? It's an enzyme that is able to, you know what it is? It's an enzyme that is able to reproduce DNA. If you make some mutation of this enzyme, you are going to promote some mutation on the DNA itself. But this is not exactly what we wanted to do. And the question asked from the first row was very interesting because we don't know exactly if the mutation are here, and we know that the mutation are here, but we don't know exactly which one. Okay. At the end of the presentation, you said something interesting. It should be interesting to delete the mutation, by CRISPR-Cas9 process. What do you think about that? Deleting the mutation. What's happened if you delete the mutation? How can you delete the mutation? You know what is CRISPR-Cas9? All of you. You cut part of the DNA. What's happened? you are going to delete all the protein. So that means that if you delete the mutation on the mitochondrial DNA, you are deleting finally the mitochondria. You know, the mitochondria DNA only codes for 13 protein. 13. That's quite low. So if you remove some parts, you are going to have maybe 12 or 11 or even less. Remember that mitochondria functioning is done by this 13 protein plus some genomic protein. Okay, this is the association between. What I have more also, yeah. So other matter to measure, this is exactly what we talk about. Yeah. The proof of concept of fixed, blah, 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 cognitive versus brain is not, maybe I can answer the question, but it's not very interesting. They measure different things about brain, but they never evaluate the cognition, if I'm not right, if I'm not true.
Yes,
So do you have an idea about how measuring cognition in this context or not? Yeah, you maybe. You are from neuroscience. You are this is on neuroscientific here, so we can ask questions.
La différence entre force?
Yeah, why maze? Why maze or X maze?
Yes.
Yeah. So I have to explain, or you want to explain? You are a neuroscientist. You know this kind of test. I mean that recurring some memories. You do the Y maze. You know what is a Y? So Y maze, okay, with two arms. One arm there is a reward, the other one there is no reward. Or maybe you can make also something that is pairing or that is totally is going to stop the animals. On the Y-axis, you have the reward, and you do that. You do what we call the training, and one day you don't put the reward, and you're going to observe if your mouse is going to remember that on the right there is a reward, on the left there is something else. That is a very simple test. You can also can do some other conditioning, but by fear. This is by rewiring. You can do by fear. You know this grid with electricity, and finally you associate it to the context, a fear feeling, by stimulating the electricity, and you are going to induce a memory, which is pain, of course, this way. The day after or 1 week after, depending on the experiment, you reput the mouse in this context, and you are just looking at the behavior of the mind. If they are what we call not shivering, but if they are, well, I don't know the word for that, freeze. If they are freeze like that, that means that they remember that this context is associated with pain. If they are not freezing, they don't remember at all because they are totally stupid and this is something. Okay, so you have tons of experiments like that. You know also the Maurice water maze. You know what it is with the platform. You know what it is? The water maze in the piscine, la piscine de Maurice. So you put the platform, and then one day, because you are a little bit, I don't know the word in English, but Cedric, you are going to remove the platform, and you observe the mice that is try to swim, but finally doesn't find the platform, but you are observing the time spent into the quarter where you put the platform before. So different tests, of course, and here this paper, one of the criticism, of course, is that they are not measuring anything else than just some brain stuff, etc. Last thing, you also mentioned something about the effect of environment. I didn't understand. What do you want to mean? And you put environment with a kind of ecologic stuff, so I don't know exactly what you mean. I'm curious. What do you think about environment of mitochondria? In French, if you want. Qu'est-ce que tu voulais dire?
C'est autour de la l'environnement par le corps.
Non, mais je trouvais que, je sais pas si c'est ça l'idée, je trouvais ça, tu sais, des fois, dans les trucs comme ça, on ne croit pas, mais il y a de super bonnes idées. Et je trouvais que c'était, je n'avais pas vu comme ça le truc, effectivement. The consequences of mutation on the microenvironment of mitochondria, because we have to think about that. Mitochondria is not, this is an organelle, but it's not selfish. It needs, as we just discussed, it needs also the genomic proteins, etc. So it could be very interesting to identify what we call the exposome of mitochondria within the cell, or for the mitochondria that are in the circulation with other part of the organism, and to see if the mutation may change their interaction with the cell or with other components. I don't know if it was your idea, but if it's the case, that was brilliant. Other comment of the question for today, it's very easy to remember what we talk about because everything was in the review. We talked about the structure, we talked about the genetic, we talked about the function. We said that some markers are already available for maybe making mitochondria at least shape as a good biomarker of aging, even if it's not validated. But in the future, there is no doubt that mitochondria should be considered to the growth production, to the shapes, to the structure, or to the function as a good biomarker of aging. We are already measuring that by mitochondrial respiration, not in clinic, not in routine. Even though that we can consider that maybe when you do the VO2 max, you know what it is, this kind of technique that measuring the capacity to use the oxygen and to produce CO2. I think that this is a reflex of your mitochondria, but globally on the body. It should be interesting in the future to get that more precisely, not at the cell, but at the tissue level, and to better understand finally the trajectory of your mitochondria. Don't think that more is bad, is better. It's not more is better for mitochondria. The fit of mitochondria is more important than is the number of the structure. The fit means that the composition of the mitochondria. We didn't have the time to talk about the different components of the mitochondria, such as the complexes, etc., and all the enzymes. You know, we never talk about the ubiquinone, about these different enzymes that are really interesting to potentiate the effect of mitochondria. We little bit talk about the strategy that are antioxidative strategy. Finally, we have also other molecules such as the ubiquinone, semiquinone, etc., that should be considered to help the mitochondria to present a better function. Okay. Just because last time I also had a question, the exam consists in few questions, maybe sometimes right or false, or sometimes you have to write something regarding the 4 different workshops. So today, last Monday, tomorrow, and the 14th. I'm not going to write a list of things that you have to know and things that you don't have to know. Okay, but that means that of course you take some notes. I hope I saw you taking some notes during the presentation. I have to correct when something is done is false, and it's not the case since Monday. So that means that, and we discussed after that about what I think that is important to retain for the future. Okay? But I totally free to pick and choose what I want about the discussion and to put that as a question. Okay? Right for you? Thank you very much for today. Tomorrow, same thing. Tomorrow it's about what? Senescence? For people that are online tomorrow, if you don't have your camera on, it's not necessary to come from me. Okay?
Doctor, sorry. Yes, is that the presentation?